Today we're going to talk about chapter 2, or this week we're going to talk about chapter 2. This is the second week. So we're going to talk about chapter 2. Chapter 2 is fairly complex. It's about the nervous system and why we act the way that we do. Let's go ahead and get started. The nervous system is composed of cells called nerve cells or neurons. Of course, you all know that. The average human body has between 100 billion and 150 billion neurons. Neurons communicate with each other at a point known as a synapse. Another cell that protects and adds structure to the nervous system is the glial cell. And there's about as many glial cells. There's at least as many glial cells as there are neurons. A neuron is composed of four major parts. The dendrites collect the stimuli or information and transmit it to the rest of the neuron. These are the dendrites right here. That's where all the information will come in to the neuron. The soma is the body. Soma always means body. It's Latin for body. So anytime you hear soma or somatic, it has to do with body. This is it contains a nucleus and it serves to maintain the cell. The axon is the main transmitting apparatus. This is the soma right here, the soma. This is the axon, and that's where the information is going. This is where it comes in. Whoops, I'm sorry. This is where it comes in. This is the part that maintains the neuron, and this is where it moves down the neuron to the end, which is known as the axon terminals. But this is the axon. It can be as long as several feet. You have a sciatic nerve that runs from the top of your gluteus maximus, your butt, all the way down to your heel. And depending on how tall you are, it can be several feet long. Mine's not nearly that long. It's not very. I'm only 5'6", so mine's not as long as somebody that's 7 or 8 feet, 6 or 7 feet. So the axon is the main transmitting apparatus. The axon terminals are where the neuron passes on the information to other cells. So the information comes in here. It runs past the soma, down the axon to the axon terminals, and that's where it's transmitted to its next whatever, or wherever it's headed. Neurons can be classified in three basic ways. You can classify them by their shape, you can classify them by their size, or you can classify them by their function. And the reality is that most neurons are classified actually all three ways. Multipolar neurons have many dendrites in a single axon. Most of the neurons of a vertebrate brain are multipolar. And as you can see, lots of dendrites. Bipolar neurons have a single dendrite at one end and a single axon at the other. Most of the neurons of the sensory system are bipolar, especially those of the retina and the olfactory system. Monopolar neurons have a single branch coming off the soma that branches in two different directions. One branch will be the input direction and the other will be the output direction, but there are axons going in both directions. Some neurons are very large and some are very small. Examples of small cells would be granule, spindle, and stellate neurons. Examples of large neurons would be pyramidal, Golgi, and Purkinje neurons, and these are found in your cerebellum. The Purkinje neurons, pyramidal cells have to do with your memory. They are in your gray matter of your brain. The gray matter is the outer, about a quarter of an inch covering of the brain, and that's where the pyramidal cells is. That's actually where most of your thinking takes place, in the gray matter, rather than the white matter. Underneath the gray matter is the white matter. Functionally, neurons either receive stimuli, create a response, or connect the two. Sensory neurons collect the stimulus and pass the message on to the vast network of connector neurons or interneurons. Motor neurons connect to muscles or glands and create the response by making the muscles contract or by changing the activity of the gland. Neurons do not touch each other, but communicate between the dendrite of one neuron and the axon of another across a gap known as a synaptic cleft. At the end of the axon is the axon terminal. The axons end in expanded areas called buttons or boutons. This is actually French for this. Buttons. The end of the axon is known as the presynaptic zone. The area of the receiving dendrite is known as the postsynaptic zone. The message is actually transported by a chemical known as a neurotransmitter that is contained in spheres in the axons called synaptic vesicles. And you can see the synaptic vesicles right here. They are holding neurotransmitter and waiting to release them into the synaptic cleft so that there can be a response. One of the interesting areas of study dealing with neurons are projections coming from the dendrites called spines, or dendritic spines. These spines seem to change with experience and have been known to change within seconds when an individual is exposed to a novel stimulus. And that's why you remember things. That's why you have muscle memory, is because of these dendritic spines, and they're constantly changing. Their mission is to adapt to the environment. And this is what the dendritic spines look like, making connections and allowing you to do the same thing over and over again or to remember something. The axon projects from the soma in an expanded area known as the axon hillock. The electrical impulse begins in the widened hillock and intensifies as it moves into the narrower axon proper. Neurotransmitters will create an electric charge. So it's an electrochemical reaction. The action of the neuron is an electrochemical reaction. Starts out as a chemical. It creates an electric charge. The electric charge runs down the axon to the axon terminal, and then it releases its neurotransmitter, and the neurotransmitter starts the whole process again by crossing over to another neuron or whatever it's crossing over to, and it starts the whole process again. And that's how it fires. And it fires it fires very rapidly. Almost all neurons have only one axon. However, some have axons that divide and form several branches. This branching is known as axon collaterals. When someone smashes their finger, the stimuli must follow a nerve pathway to tell the brain what has happened. Nerves that take stimuli from its point of origin to the central nervous system are called afferent neurons. When the brain or spinal cord sends back a message to the affected area, the neurons on the reply pathway are referred to as efferent neurons. Afferent and efferent. It's kind of interesting that they are almost the same word. Who came up with this? Anyway, so if you think of it alphabetically, the information is going to the central nervous system with the afferent neurons that start with an A, and they're coming back to the area from the brain to the affected area by the efferent neurons. E, it's alphabetical. A, E, there you go. So that's one way to remember it, I guess. The soma manufactures proteins that maintain growth and function in the neuron. This protein is able to make its way down into the distant areas of the axon and dendrites by a process known as axonal transport. The protein is moved along tiny hollow cylinders known as microtubules. Protein molecules travel down smaller rods inside the microtubules called neurofilaments and even tinier microfilaments. Now you may ask yourself, well, why in the world will I care about this? And the answer is because some of our pharmaceuticals, the reason they work is because it either blocks the transport of select proteins, or or it accelerates the proteins. And that's why some of your medications work. So this is really very important. It's very important that we understand these things as we are treating people, or if we are being treated with select chemicals that block something. I'm on blood pressure medicine, and what happens is it's a beta blocker, and since it's a beta blocker, it slows down the protein processing in the neurons, and it slows down my reaction to things. Glial cells are part of the neuronal system in that they provide structure to the neuronal groupings and help hold them in place, and they help maintain the neurons by feeding them. There are 4 types of glial cells: astrocytes, microglial cells, oligodendrocytes, and Schwann cells. The astrocytes are star-shaped glial cells that protect neuronal bundles, especially in the brain. The dura mater, a tough sheet that is wrapped around the brain for protection, is composed of astrocytes. Astrocytes contribute to the metabolism of the neuron and provide restorative protein to them. So if you ever are part of brain surgery, the first thing that they will have to do after they open the skull is to cut through the dura mater, or this layer of astrocytes. Kind of interesting. Microglial cells are very small, hence the name. They migrate to areas of injury or disease and remove debris from injured or dead neurons. Now, this is important. As you get older, you're losing more and more neurons in your brain, and these need to be cleaned out. If they're not cleaned out, then it can cause Alzheimer's disease. And so the microglial cells, that's their job. They think possibly it's a malfunction of the microglial cells that causes people to have dementia and Alzheimer's disease. Myelin sheaths that surround and protect the neurons are created by glial cells. One of the interesting things about microglial cells, you never know what's going to happen or what your genetics will tell your microglial cells to do. I have a friend whose father died in his 40s from Alzheimer's disease. It was rapid onset Alzheimer's disease, and it was basically a problem with his microglial cells. And of course, we have discovered this over time. This was back in when we were in college. So that was 50 years ago. He was afraid to have children because he was afraid that his children would have rapid onset Alzheimer's disease like his dad did. So he didn't have any children. His sister did, but he didn't. None of them have it. He was afraid he had it, and he's still alive. So I'm 72. I think he's 72 as well. So he dodged that bullet, that genetic bullet. Anyway, but one of the things that you can do, you can really accelerate your aging process by taking toxins into your system. So people that drink have been alcoholic, potentially they will develop one of these dementias in their 70s or 80s. Somebody who smokes tobacco, it's another toxin that can cause these kinds of problems. So one of the reasons that we're not exactly sure what's going on is because back in the 50s and 60s and 70s, there were a lot of people that smoked, and they drank a lot, and they smoked a lot. We don't have any clue what marijuana does to these processes, but potentially, if you stay away from these things, you can live into your 90s and even into your hundreds without having to worry about developing Alzheimer's disease. Just a thought. We're still not sure exactly how this whole process works, but we do know that people that are alcoholic, people that drink a lot, people that take drugs, put all these toxins into their system, they're more likely to develop Alzheimer's disease and all the other problems. and a lot of it has to do with the insults they've made to their brains, um, especially cocaine, amphetamines, people that use crystal meth. These individuals are accelerating the process. They are stopping a natural process that takes place so that they can be manic. It creates a manic situation, but it's also toxic for these nerve cells in your brain. You can fry your brain if you take too much of this stuff, but even if you don't fry your brain, then potentially the toxins that are there can cause recurring problems. This is just something that you need to think about. We will find out more about this as time goes by. A lot of the problem has been with research that people who drink don't like to be told negative things about alcohol. People that smoke pot don't like to be told negative things about marijuana. People that smoke, of course, tobacco companies controlled the science of tobacco for an extended length of time. And one of the problems has been that we weren't getting very good information. I've got a call. It's my wife. Just a second. Hello? Okay. All right. Okay. All right. Thanks. Bye. Yeah. Okay. Bye. Sorry, that was my wife. Okay. Anyway, so this is just something to think about. We'll be getting more information as time goes by. Probably not very good information from marijuana because there are too many people and people want to sell it and they don't want to get it outlawed again. So they probably the good information. It's going to take a while to get that information. We already know about alcohol and tobacco causing problems, and cocaine, of course. Um, Robin Williams had, what did he have? He had a form of Alzheimer's disease that was rapid onset, and that's one of the reasons why he committed suicide. But Robin Williams, in his younger days, was a cocaine addict, or he used a lot of cocaine, and of course he may have damaged his brain doing that. Made him talk really fast, and he was pretty funny. Myelin sheaths that surround and protect the neurons are created by glial cells. These myelin sheaths not only protect the neuron, but accelerate the response of the neuron. Oligodendrocytes provide the myelin in the brain and spinal cord. Oligodendrocyte means little tree cell because it looks like a little tree. One of the problems we have with celebrities, they come down with these problems. They die at 59, or somebody just developed or admitted that they have multiple sclerosis. Oh, it's Christina Applegate. The problem is that if they told us what they did when they were younger, the things that they did, like yeah, I snorted a lot of cocaine when I was a kid. Robin Williams actually said that, or when he was young, he actually said that. But if we got that kind of information, then people would be more aware that yeah, geez, maybe I shouldn't. Yeah, I thought this was cool back in the day, but now I know that if you do this, then it may accelerate your life and give you some problems. And of course, we don't have people telling us that, and that's part of the problem. We'd be better if we had that information. Schwann cells construct the myelin outside the brain and spinal column. Schwann cells are small, and each one will wrap itself around the axon to protect it and accelerate response. When you look at the myelin formed around an axon, you will see that Schwann cells don't touch each other but leave a gap between each cell. These gaps are known as nodes of Ranvier. When an individual suffers a blow to the head, the injury tends to heal slower than a blow to any other part of the body. This is because, besides causing localized damage, the glial cells become larger by taking on moisture. This is known as edema, and it may put pressure on the select parts of the brain, causing malfunction. And the reason this is done is to protect these cells. If every time you got your head hit, it destroyed lots and lots of cells, then pretty quickly you would never make it out of adolescence with an intact brain. But what happens is the glial cells will expand and protect those neurons, and so fewer of them are killed. And this is one of the reasons why you get so much swelling in your brain if you get a really severe blow to the head. So, this is one of the reasons why people will go into comas and then they'll come out a couple weeks later after the glial cells have decided that the trauma is gone. Then the edema will go away, the swelling will go away. But they're there just to protect that body part. Let's assume you're walking along and you sprain your ankle. All of a sudden, it just turns into a balloon. You've got this balloon around your ankle, and you're thinking, "Hey, I don't need a cast. I've got a internal cast. I don't really need one." Well, that's the whole reason that your ankle will swell like that is to protect you, and you could probably function a little bit better. You could walk, not very well, but you could still walk. I mean, you're not completely crippled by your sprained ankle, uh, and that will get you out of trouble. And of course, eventually it will go down, and now and it will heal. Tends to heal a lot faster. Muscles tend to heal faster than brain tissue, and that's called edema. And we try to control edema. Anytime we have a infectious process or a traumatic process anywhere in the body, we will see edema. This can be a problem if the damaged area is your heart. The edema will build up around your heart, and of course that can cause congestive heart failure. That and the fact that your heart is not functioning properly, and you're not getting rid of your fluid from your lungs, so your lungs will fill up with fluid. But you can also get that edema around your cardiac sac. That not a good idea. Anyway, the nervous system of the human body can be subdivided into two nervous systems. The central nervous system is comprised of the brain and spinal cord. The peripheral nervous system includes all other neurons outside the brain and spinal cord. And this is the peripheral nervous system right here. As you can see, it's everything except the spinal cord and the brain. And there's the central nervous system, the spinal cord, and the brain. The peripheral nerves are categorized by what part of the central nervous system they are connected to. Cranial nerves are connected directly to the brain. Spinal nerves are connected directly to the spinal column. Autonomic nerves in the autonomic nervous system are connected to glands and organs and trigger automatic responses from these areas. There are 12 cranial nerves that control sensory or muscular movements in the head or the neck region. 4 of the nerves control the muscles of the eye and eyesight sensations. 4 control the muscles of the neck, face, and tongue, and the sensations of the tongue, sinuses, and throat. 1 controls the sensations of the inner ear. The vagus nerve controls the heart, lungs, and your digestive system. And these are your 12 cranial nerves. There are 31 pairs of spinal nerves, 1 pair on each side of the body. There are 5 segments of the spinal cord and column. There are 8 cervical nerves. This cervical means collar. That's in your neck. Collar. I do not pronounce that correctly because I am from Indiana, and people from Indiana can't pronounce collar. They get it mixed up with a lot of other different words like collar, Color. Anyway, that's one of my problems. There are 12 thoracic nerves. This is in your torso. Thoracic means brace. There are 5 lumbar nerves. This is in your lower back. This is where a lot of people have problems. This is right around your belt region. It does mean belt. Lumbar means belt. There are 5 sacral nerves. That's your girdle. That's around your pelvis. in your pelvis area, and sacral means girdle. And there's one coccygeal nerve that's in your butt. It's right at the end of your spinal column, and it means tailbone, coccygeal. Anyway, all these words are Latin, and they all have meaning. So if we were in ancient Rome, of course, somebody would talk about the cervical nerves, and we know that it's around your neck, and we know that thoracic means brace, which is in your torso, and we know that lumbar means belt, so it's around the belt, the area where you have a belt, and sacral means girdle, and you'd know that it was between your belt and your thighs, I guess. And then coccygeal, nobody, that's not a very common Latin word, but the other four are all common Latin words, and there they all are, all the nerves we're talking about, and you can see which nerves actually control things. The cervical nerve is really kind of interesting. If you have a problem with your cervical nerves, it can cause a lot of problems. If it's down low in your cervix, that cervical area, not your cervix, your cervical area of your neck, can cause a lot of problems. So this is one of the reasons why whiplash can cause problems, a lot of serious problems. Now, as you can see, there's a natural S curve, to your cervical nerves, to your cervical vertebra. What happens with whiplash is that this vertebral column gets straightened out, and that's not the way it's supposed to be. It's supposed to have a natural curve in it. If it doesn't have that curve in it, now you got a problem. You can have a problem with any of these things. It can potentially cause a breathing problem. Your arms can become weak. Your deltoids are the points of your shoulder muscles, and your biceps, of course, are the front of your upper arm muscles. Wrist extenders, you can start having interesting problems in your wrist. Triceps are a little bit lower. That's the back of your upper arm muscles, and of course, hand. And this is one of the reasons when somebody has an accident, we try to get them to respond, to extend their fingers, to move their wrists, to check their arm muscles. Well, actually, this is one of the ways the doctors do it. If you come across somebody with a possible neck injury, don't move their neck, because potentially you can do damage to any of these nerves, and then that will cause all kinds of interesting problems. Anyway, we won't talk about that anymore. The autonomic or automatic nervous system is divided into two nervous systems: the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system prepares the body for action by shutting down normal functions, increasing power stores, and increasing sensory activity. The parasympathetic nervous system returns the body to a normal state after the sympathetic reaction. When the need for action is detected, the sympathetic nervous system will dilate your pupils, improving your eyesight. Excuse me. It inhibits salivation, causing dry mouth. It relaxes your airways, allowing deeper breathing. It accelerates your heart. It stimulates your sweat glands, giving you what they call flop sweat. It stimulates liver to release energy stores. It slows down your digestion. It inhibits your kidney function. It constricts your blood vessels in your skin, making you look paler than you normally are. When people say they can smell the fear on you, this is what they're talking about. Your sympathetic nervous system is kicked in, and you've got this flop sweat going on, so that you have a different odor about you. And you can detect this. Really kind of interesting. I worked in medicine for 30 years, and a lot of times I saw this when people were afraid. You could detect it with their smell before you could detect it by looking at them. Of course, if you look at them, then they look pale. They're sweating from interesting areas, especially their forehead. they're working their mouths because they can't get any saliva into their mouths, and they're probably breathing a little bit differently than they were before. But you can also look at their pupils and find out if their eyes are dilated or not. Parasympathetic nervous system kicks everything back into normal reactions that constricts the blood, the pupils in your eyes. It stimulates salivation so that you're drooling instead of you can't get enough saliva in your mouth. It constricts your airways so that and this is one of the reasons after a trauma is over with why people breathe funny. They have an extended problem with this. It slows your heartbeat, it stimulates your digestion, it dilates your blood vessels in your intestines. It dilates blood vessels in the skin, and the individual will look flushed. So you can just look at their skin, and you can tell whether the sympathetic nervous system is kicked in, or whether the parasympathetic nervous system has kicked in. A quick story. When I was in college, I ran track. One weekend, my wife and I were arguing about something, and she was going to leave me. On the following Monday, I had a track meet. I ran track when I was in college, and that morning I had student teaching, which is, you always get nervous student teaching. Anyway, so I had a bad weekend with my ex-wife. Well, she's my ex-wife now. She wasn't then. I had a bad weekend with her. The argument started on Saturday, and by Sunday she had loaded up the car and she was ready to go. So I started to take her home, and about halfway she decided that she didn't need to leave me. I can't remember what the issue was. There was something going on. It didn't have anything to do with me. It had to do with her uh, not being happy where she was. Anyway, she came back home, and we were okay. Tough weekend though. I'll tell you, it was a tough weekend. So Monday morning I got up. You had to get up early, like 6:30 in the morning to go and to student teach. So I student taught until noon, and then I went back to campus and I took classes all afternoon. And then at 4 o'clock, the track meet started, and I used to run I ran the 440 relay, the 440, the 220, and the mile relay. I ran all those races, and by golly, I wasn't running very well. I was slow. I think I came in third. I came in second in the 440, and we won the 440 relay. But boy, my leg was not that fast. My leg of the relay was not that fast. I came in second in the 440. I came in third in the 220. Um, and I threw up after every race. And so by the time the mile relay came along, I couldn't keep anything down. And they replaced me. They replaced me. I can't remember who they replaced me with, but we won the race and we won the meet by like 2 points. And part of it was my fault because I didn't score any points. So I'm still throwing up, and this is after the meet's over with, and they think, "Oh my God, he's got the flu." So they took me to the hospital and they checked me out, and what they discovered was that wait a minute, that's a parasympathetic nervous system. What they discovered was that my kidneys weren't working. And the reason my kidneys weren't working was because of adrenaline. I had hyperadrenalinemia. At that time, that was just after Joe Frazier and Muhammad Ali had fought the Thrilla in Manila. And it looked like Joe Frazier was going to beat Muhammad Ali. And then in the late rounds, he kind of lost his he lost it in the last rounds, and it went 15 rounds, and Ali was named the winner, but afterwards they checked out Joe Frazier and they discovered that his kidneys weren't functioning, and the reason was because of hyperadrenalinism. and they started calling it athlete's kidney, which is situational kidney failure is what they referred to it as. So I was in the hospital for a week, them trying to get my kidneys. My kidneys were working, but really slowly. And of course, I had to relax, and the food was horrible, and all the rest of it. Anyway, I had my sympathetic nervous system kicked in on Saturday when my wife started giving me such a hard time, and by Monday I was in pretty bad shape. My kidneys weren't functioning very well, and you need your kidneys to clean out. Now, this is one of the reasons why diabetics need to go on dialysis, because they need to clean the toxins out of their system. That was an incident where my kidneys stopped functioning. And it was due to my sympathetic nervous system hyperadrenalism, or in my case, they referred to it as athlete's kidney. The average human brain weighs about 1,400 grams, about 3 pounds. The major part of the brain is the cerebrum, which is divided into 2 hemispheres. Intellectual capacity seems to be contained in the surface area of the brain, as I was telling you before, the gray matter that is on the surface of the brain. Surface area of the brain is increased through ridges and folding that triples the surface area of the brain. And these convolutions are called gyri, and the furrows are called sulci and gyri. The brain contains 4 fairly distinct lobes that have 4 fairly distinct functions. The frontal lobe is in the front of the brain and is the seat of higher level thought. And this is the front of the brain. The parietal lobe is on the top of the brain and controls fine body movement, and that's the parietal lobe. The occipital lobe is in the back and controls sight. That's the occipital lobe back here, the green portion. And the temporal lobe is on the side and controls hearing. And this is one of the reasons why when you get hit in the head, you have one of the things we do is we'll ask you questions. And what we're trying to do is we're trying to determine if any portion of your brain is damaged. If you have poor eyesight, it's one of the reasons why we hold up a number of fingers and ask you how many fingers am I holding up. We're trying to find out how good your eyesight is. If you can answer us, it probably means that your temporal lobe is, if you can hear us, it means your temporal lobe is damaged, and it also means your occipital lobe is functioning properly. If you have a hard time moving around, that of course would be your parietal lobe. But people rarely get hit on top of the head. Normally they get hit in the front of the head, the side of the head, or the back of the head. And actually this brain is sitting there on a stalk inside your skull, and there's a mass of fluid around it, and this is your cerebrospinal fluid. So if you get hit in the front of the head, you not only hit where you're hit in the front, but it also rebounds, and you get a blow to the back of the head at the same time. So this is one of the reasons why we ask you all the silly questions that we ask you. Who's the President of the United States? What day is it? And what we want you to do is answer us, because if you can't speak, it means you have damage to your frontal lobe. That's especially the left side of your frontal lobe if you can't speak. If you don't understand what we're saying, that means that you have damage to another area, an area that's right here. And you can't process information that you hear. So that's one of the reasons why we ask you all the silly questions after you get hit in the head. If an individual lost half of their brain before they reach puberty, except for some fine motor movements, they would seem perfectly normal. They only have half a brain, yet their intellectual capacity is the same as it would have been. The 2 hemispheres of the brain are connected by a mass of thick myelinated neurons called the corpus callosum. And this is a corpus callosum, this red thing right here. It's not really red in your brain. It's actually white. It's very pale. The rest of your brain is kind of an off-white, and of course the gray matter, but this portion is white. This is connective tissue, and it connects the 2 hemispheres of the brain. Now the interesting thing about the corpus callosum is you can damage this thing. Women who drink during pregnancy uh, when the corpus callosum is forming in their fetus, this will not form properly. And when we talk about fetal alcohol syndrome, I'll show you the difference between a normal corpus callosum and one that is affected by the alcohol that the individual drank. The interesting thing about the corpus callosum is that women have a larger corpus callosum, about 11% larger than the male corpus callosum. And this really changes some of the dynamics of what happens to you if you ever get sick, especially if you have a stroke. And we'll talk about that later. If we cross-sectioned an individual's brain, we would see that some of the brain is made up of tightly packed non-myelinated neurons that look gray. Other areas look white because of the myelinated neurons that make them up. Researchers have found that the larger the gray areas of the brain, the more intelligent the individual is. I was looking at this when I was preparing for this semester, and I was reading articles about the damage that marijuana does to the brain, and this is one of the reasons that people who smoke marijuana, um, their intellectual capacity may be damaged. Their memory certainly is damaged, but the reason is because marijuana um, makes the gray matter smaller. People that have uh, who smoke who are chronic marijuana smokers have a smaller gray area than people that don't smoke marijuana. As interesting as that is. You can look at the statistics. It's really kind of fascinating. Most of this research, we haven't been able to do this in the United States, but most of this research comes out of Canada and New Zealand. And the reason is because they decriminalized marijuana usage way back in the 70s. So a lot of people in Canada and New Zealand have been smoking pot for an extended length of time, for 25 or 30 years. So we're getting a lot of good information from those two places. The United States haven't done a whole lot. It's been illegal, so you couldn't really do research with it. So you can take that to the bank, I guess, if you like. We just need to think about what it can potentially do to you. As the baby begins to develop from ever-expanding numbers of cells, the brain and the central nervous system begin as a structure known as the neural tube. In the beginning, the smaller, more primitive parts of the brain develop before the intellectual portion of the brain, the cerebral cortex. The spinal cord will develop medulla, pons, and cerebellum. All of these develop long before the cerebral cortex develops. But the baby's brain is developing the entire time that the baby is in the womb, for the whole 9 months that the baby's in the womb. And this is one of the reasons why doctors tell women not to drink, because the alcohol has an affinity for brain tissue. Strangely enough, and what a surprise! Those of us who have ever been drunk know, yeah, it's got an affinity for the brain tissue, all right. But it's doing the same thing in the fetus's brain, and it's constantly being built. So even binge drinking, of course, is the worst for a developing fetus. But any degree of drinking at any point during pregnancy can do damage to the baby's brain. The cerebellum surrounds two areas that scientists consider an evolutionarily old part of the brain that they sometimes refer to as the reptilian brain. There are two portions of the reptilian brain: the basal ganglia, which means basic nerve sheath in Latin, and limbic system, which means border system. The basal ganglia includes the caudate nucleus. Caudate means tail. Putamen, which means stone. Globus pallidus, which means pale sphere. And substantia nigra, which means black substance. And the globus pallidus, as you can see, is pale, and the substantia nigra, which isn't here, are actually black spots, as curious as that is. The limbic system is made up of the amygdala, which means almond. It's shaped like an almond. The amygdala. Where are you, amygdala? Oh, there it is. That's the end here. the end of the hippocampus. The hippocampus, which means seahorse, looks like a seahorse. The fornix, which means arch, and the fornix, of course, is an arch. Cingulate gyrus, round girdle, and cingulate gyrus is here, structure right here. And olfactory bulb, where are you, olfactory bulb? Oh, it's not on here. And the mammillary bodies, these are the mammillary bodies. The largest organ in the center of the brain is the thalamus. Thalamus means inner chamber. Thalamus acts as a connector between the upper and the lower parts of the brain. The real controlling gland in the brain is the hypothalamus, and the hypothalamus is right under the thalamus. Hypothalamus, of course, means lesser thalamus. When we first cut into the brain, we had no idea what their functions were, and when they found the thalamus, they thought, ooh, this must be a controlling portion of the brain. But actually, it's just a conduit. It acts as a transfer station for information. The hypothalamus is called the hypothalamus because it is below the thalamus. It's known as lesser thalamus. It's right there. In reality, the tiny portion of the brain controls your hunger. It controls your thirst. It regulates your temperature. It controls reproductive behavior. It controls the pituitary gland, which controls almost all of the body's hormone secretions. So as you can imagine, if you damaged your hypothalamus, you'd be in a lot of trouble. I had a friend who had cancer when he was 14 years old, developed cancer when he was 14 years old. And while they were trying to eradicate the cancer, they cooked his hypothalamus with radiation. They destroyed his hypothalamus. Every day he had to take a whole handful of drugs to control all of these things because his hypothalamus was not completely functional. And because of that, he had to take all kinds of drugs to control the hormone secretions in his body. Two of the most important areas of the midbrain are the tiny bumps known as the superior and inferior colliculi. Colliculi means trough. The superior colliculi receives visual information. The inferior colliculi receives information about sound. The substantia nigra, the black substance, releases dopamine, and when the substantia nigra is damaged or it starts to decrease, it causes Parkinson's disease. And the reason is because you're not releasing enough dopamine, and not enough dopamine causes Parkinsonian symptoms, tremors, and difficulty walking and whatnot. The reticular formation runs from the midbrain into the medulla. This portion of the brain controls sleep and arousal, temperature regulation, and motor control. It is a reticular formation that arouses a person when their baby cries. And as you can see, it is quite a network of neurons. If you do damage to this area, it's going to put you into a coma. And the reason is because if you have swelling against your reticular activating system, then if you have damage to that, then it puts you to sleep. And of course, you can't walk. You're in a coma, and you have a difficulty maintaining your body temperature. The cerebellum is composed of tightly packed and folded neurons. The neurons in the cerebellum form an interesting fan-shaped structure that includes granule cells, Golgi cells, and Purkinje cells. And these are the very intricate cells we were talking about. They're large. They tend to be large cells. Purkinje cells are contained in the middle layer of the neuron in the cerebellum. Purkinje cells are large, multipolar neurons shaped like fans with many dendritic spines. These cells run from the surface of the cerebellum to the brainstem. The intricacy of the Purkinje cells allows the cerebellum to control both the fine and gross motor functions of the body. The position of the cerebellum between spinal cord and thalamic centers that communicate with the motor complex allows it to control motor function. And for the longest time, we thought that was the only function of the cerebellum. but now we know that it also has to do with memory and it has to do with complex thought. Not as much as the frontal cortex, but it still has to do with both. The cerebellum leads into the pons, the first portion of the brainstem. The pons, which means bridge, is involved in motor control and sensory analysis. Information from the ear first enters the brain in the pons. The bottom of the brainstem is made up of the medulla, marrow or middle, which is what medulla means. The medulla controls both the neck and the tongue muscles. The medulla also contributes to the regulation of breathing and heart rate. Pons, I used to live in Zweibrücken, Germany. The French name for Zweibrücken was Dupont, which means two bridges, because there were two bridges across I don't think it was the Rhine River. I think it was the Rhone River. Anyway, whatever river it crossed, there were two bridges, and they were right there in Zweibrücken, two bridges, or Dupont, which means two bridges as well. So the DuPont family comes from Zweibrücken. They come from the Zweibrücken area or the DuPont area. And that's just an interesting piece of side information. The main intellectual functions occur in the cerebral cortex, which means external layer. The cerebral cortex, the gray matter, is 6 layers thick and contains between 50 billion and 100 billion neurons. The functions of the various regions of the cerebral cortex have been mapped and divided into 46 distinctive areas known in toto as Brodmann's area. Why? Because Brodmann was the guy that discovered it. The most numerous neuron in the cerebral cortex is the pyramidal cell. Pyramidal cell dendrites reach to the surface of the cortex and also spread out horizontally. These neurons seem to be arranged in columns, and this is one of the reasons why if you ever watch Jeopardy and try to answer the questions, sometimes you can answer them and sometimes you can't. But the reason that they can ask you a question and you can come up with the answer is because of this horizontal movement or this horizontal position of the dendrites of the pyramidal cells. So you had theoretically, if you've ever heard this piece of information before, it starts out here, and you can connect it to other portions of this cell very rapidly, and then of course you can say what the answer is, or push on the button. I guess in Jeopardy, that's what you do. Many brain regions have distinctive geometric columnar patterns that seem to function as information processing units. Some columns begin at the surface and extend all the way to the white matter. The human cerebral cortex contains about 1 million cortical columns. Most cerebral communication runs vertically, but there are some areas of horizontal communication. So anytime you're asked a question and you need to come up with the answer, you're thinking both horizontally and vertically as far as your pyramidal cells are concerned. Now, all of these have to be arranged correctly, of course, in order for that information to come out. And most of us, that is the state of our brains. But some individuals, their pyramidal cells are disorganized. And we'll talk about that when we're talking about schizophrenia. And there we go. The cortical column of the neocortex are arranged in 6 distinct layers. However, most communication is vertical, and select neurons will extend through several layers, some through all 6 layers. And this is the information that we're looking for. And as you can see, this is what the layers look like. Besides the bony skull, the brain is protected by 3 protective sheets called meninges. Meninges actually means membrane. Meningitis is an infection of the meninges. The outer layer is a tough envelope of cells called the dura mater, which means hard mother. I guess you can take that any way you want. The middle layer maintains the open portion known as the arachnoid, through which flows CSF, and CSF is cerebrospinal fluid. It is a clear liquid that is very similar to the serum in your blood. It's salty. If you ever taste it, please don't. You'll probably never come in contact. Hopefully, you'll never come in contact with it. People have a blow to the head that is fairly severe, then potentially cerebral spinal fluid will leak from different areas of their body. Usually, it's mixed with blood, but that's nothing we want to think about at this point. The inner layer is a delicate membrane known as the pia mater, or gentle mother. So there's three layers: the hard mother, the gentle mother, and the arachnoid area in the middle between the two. And the dura mater, as we said before, are made up of astrocytes, and they really protect the brain. Normally, this isn't what gets infected with meningitis. With meningitis, it's the gentle mother, it's the pia mater that becomes infected. Um, the human immune system is quite amazing. Very rarely will anything get into your brain, but if a bacteria happens to get into your brain, it can kill you fairly quickly. And one of the reasons is because if this swells up, if the pia mater swells up, it can damage your brain, and that's something that you don't want. Um, Usually, if you have a bleed into your brain, they can draw that off. It's localized, but this is general. And of course, I have some horrible stories about meningitis that I will try not to tell you. The brain is not a solid mass. There are four open areas called ventricles. Ventricles 1 and 2 are lateral ventricles in each hemisphere. As you can see, they are here. Ventricle 3 is between the hemispheres below 1 and 2. This is it right here. And then ventricle 4 is in front of the cerebellum, and this is ventricle number 4. So this is 1 and 2. This is 3, and this is 4. CSF, cerebrospinal fluid, is produced in the choroid plexus, which means separate braid, a portion of the lining of the ventricles. The only reason I'm telling you this is because people read these scientific names and they think, oh my God, where did they ever come up with that? The answer is it's all in Latin. So if we were, like I said, if we were in Rome, we would understand all of these things just from the words that we would be reading. But because we are so far removed from my Latin background, not yours, you don't have a Latin background unless you have Western ancestry. Then if you're far removed from this, these are all alien words to you, which they may be anyway. But the cerebrospinal fluid is produced in the choroid plexus, a portion of the lining of the ventricles. CSF is basically plasma or blood with the red and white cells removed. The functions of the CSF include acts as a shock absorber for the brain. It's a liquid envelope that surrounds the brain. The brain floats in cerebrospinal fluid, so head movements do not disturb the brain. You can move your head all you want, and probably it won't bother you. It collects nutrients from blood vessels and passes them on to the brain surface and keeps the brain functioning properly. That's what the CSF is for. One of the things that I have had to do, I was a laboratory technician, so one of the things that I had to do was I didn't collect the cerebrospinal fluid, but I had to analyze the cerebrospinal fluid. There's not that much of this stuff, so you don't want to pull out too much of it. Your body will continue to manufacture it if it runs low, but you can give somebody a pretty good headache by drawing out too much cerebrospinal fluid. They do that through the lumbar region of the back of the vertebra. And if they make a mistake, they hit a blood vessel, then you get blood in the cerebrospinal fluid. There's one of the things, if somebody has meningitis and you look at their cerebrospinal fluid, it's cloudy because it has bacteria in it. Normally it's clear as glass. You can look right through it. Clear as water, I guess. But if somebody has an infection or somebody has hepatitis, it'll turn yellow. So you can draw off somebody's cerebrospinal fluid and get a lot of good information from what's going on in their central nervous system. Blood is supplied to the brain through two significant arteries that run on each side of the esophagus. They're carotid, which carotid means plunge into sleep. The carotid arteries, the vertebral arteries bracket the spinal cord and enter at the base of the skull. Okay, we won't go into that. Okay, there you go. As you can see, there they are. There, here's the carotids, and here's the what was the other one? Vertebral arteries are back here. Are the blue ones? There you go. Here you just see them. You can see them in uh, this picture. At the base of the brain, of the carotid and basilar arteries join to form a structure called the circle of Willis. The emerging of these two cerebral arteries provides a backup in case blood flow is impinged in one of the other two major arteries, and those are your carotid arteries or your vertebral arteries. One of the interesting things is I have a friend from Montana who had wasn't a clot, it was a blockage in one of his carotid arteries. He didn't know it. He couldn't stand up real fast, or he would sometimes feel dizzy. But his problem was that his carotid artery was partially blocked. So they went in and they cleaned it out, and he's fine. But he was okay anyway because of these alternative blood flow routes going into the brain. Despite the rich tissue of the brain and the myriad of viruses and bacteria seeking entry, infections in the organ rarely occur. This is because the capillaries in the brain are much smaller than the capillaries in other parts of the body. Substances in the blood will rarely pass into the brain. This is called the blood-brain barrier. An angiogram, that's what we saw before. That is an angiogram. Is an x-ray of the blood vessels. If a stroke is suspected, an individual can be injected with a dye, and the skull can be x-rayed to show possible hemorrhages, aneurysms, or occlusions. Occlusions are clots or blockages, and aneurysms are widening areas in the brain. This is showing a blockage in this portion. He's got a blood clot right in here somewhere. This is usually the first test performed if a patient is suspected of having a stroke. There's a reason why they do this. They can give you a shot that will break up clots. Now this is good if you've got a clot. You can take this shot. When I had my heart attack, they gave me a shot of this stuff because they were afraid that I would have a stroke before they could take care of me. This was in Montana, and I was about 150 miles away from a hospital that could deal with me. So they gave me this shot before they threw me in the helicopter and flew me down there. Anyway, they gave me the shot. The problem is that if it's a hemorrhage or if it's an aneurysm, it'll make you bleed out, and of course, then that will kill you. So they have to be really sure this is a blood clot. About 70% of all strokes are due to thrombosis to clots. So it's a pretty good bet, but if you're wrong, 30% of the time you're going to be wrong, and you're going to be killing the patient instead of saving the patient. And we'll talk about this later on. A CT scan is an X-ray of a thin sliver of tissue. If a patient is suspected of having a tumor or a stroke, a CT scan would allow the physician to visualize the affected area, and that's what we have right here. That is not a tumor, that's a stroke. That's a hemorrhage. It's not a clot, it's a hemorrhage. He's bleeding into his brain. Whereas the angiogram and the CAT scan are both x-rays, the MRI uses radio waves and other magnetic energy to visualize the structures of the brain. Small changes in structure, as small as the myelin on individual axons, can be visualized. PET scans are machines that detect radioactive activity in the brain. The radioactivity comes from radioactive impregnated glucose injected into the subject. As the various areas of the brain metabolize the glucose for their activities, the researcher is able to see which areas are active with select tasks. This is somebody watching a nature video, and this is somebody who's a cocaine addict watching. Or I guess it's the same guy. He's watching a cocaine video. You can see how much more excited he gets from watching the cocaine video. A functional MRI differs from the MRI machine in the intensity of the magnetic signal that is used. Because of the functional MRI is so powerful, it can detect minute differences in metabolism. This gives researchers a similar picture as the PET scan, but without the radioactive injection. PET scans and functional MRIs are fairly expensive. I can't remember what that is. Okay. Anyway, so these are functional MRIs. As you can see, they've cut out a portion of his brain. This is post-surgery. This is the tumor, and that's the area that they've taken out. There you go. Okay, that's the end of the chapter. I will talk to you guys next week. Next week we tackle chapter 3. So talk to you next week. You can turn in your article critiques. There is a video about how you do them. It's not very good. Well, maybe it is. Maybe it's okay. Anyway, I'll see you guys next week.